Hello, Parish Virtual Honors Biology 2020. I have made you this Chapter 9 review because I feel bad that we really are covering a lot of information very fast and it's all virtual. And normally when we're in the classroom, I can give you chocolate, and cookies and donuts, and it really helps to learn the information while eating donuts, etc. So I am making this review and giving you some example questions and trying to frame the information because it really is a lot of information. So very, very, very sorry about that. So Chapter 9, as we talked about in class, covers cellular respiration. And if you boil it all down, cellular respiration is going to take energy from food and put it into a molecule that we can use right now. And we discussed that ATP is the perfect molecule to provide energy quickly, and the ATP molecule can be regenerated to produce energy again. Cellular respiration is really just regenerating ATP from ADP. And it seems like that wouldn't need so much to occur inside of a living organism. However, it's quite complex and complicated. In this image that I got from the internet, shows you how ATP is regenerated. A phosphate group will be removed from ATP, and then that phosphate group will bind to a molecule in a reaction, and then it helps move things along to get that reaction to have enough energy to get over the activation energy and the energy of the transition state that we talked about in Chapter 8. And that's awesome. However, that ATP can be reused. So during cellular respiration, all of these ADP molecules get rephosphorylated, and then they can go catalyze, well, not catalyze, but it can go provide energy for other reactions to occur. So in these orange boxes here, on the right, this is the process where we're actually using the ATP right this very second. And then the orange box on the left describes how cellular respiration will regenerate ATP. There are two categories of reactions that will rephosphorylate ADP that we're going to talk about. There's others, but for cellular respiration, we're going to discuss substrate level phosphorylation and oxidative phosphorylation. So those are the two ways that ADP is rephosphorylated during cellular respiration. For this class and for this test, you don't need to know a ton about these processes, but you do need to know where they occur and how much ATP is produced. So substrate level phosphorylation takes place during glycolysis and during the citric acid cycle. And oxidative phosphorylation takes place during the last stage of cellular respiration, and depending on the textbook you're using, the textbook might call this last stage oxidative phosphorylation. Sometimes it's called just the electron transport chain, but the more advanced classes that you take, they're going to call it oxidative phosphorylation. And substrate level phosphorylation occurs in glycolysis in the citric acid cycle. And substrate phosphorylation is going to produce a very small amount of ATP in comparison to oxidative phosphorylation. And substrate level phosphorylation only involves, for the actual phosphorylation of ADP, there's going to be a lot of reactions that lead up to it, but the actual step of phosphorylation only needs this enzyme and the substrates, and then that's enough. We're going to find out that oxidative phosphorylation takes a lot of other steps for it to occur, but substrate level phosphorylation is the removal of a phosphate group from a substrate, and then that phosphate group binds to ADP. So I found this really good image on the internet, and the image on the left is a very, generic, lower level comprehension image. But you guys are so smart, I included something that looks a little more like you would see in organic chemistry, where you start to focus on which electrons in which orbital from which atom are actually participating in bond formation and bonds breaking. So that type of image is in your near future. Now, oxidative phosphorylation is the last stage of cellular respiration. So substrate level phosphorylation happens at the beginning of aerobic respiration, and then oxidative phosphorylation is at the end of cellular respiration. This image comes from the Khan Academy, their website, and I really like their images from their website. So I added it here. So this is all of oxidative phosphorylation. And we talked about in class that this occurs in the mitochondria, and the electron transport chain is the first part of oxidative phosphorylation. And then chemiosmosis is the second part of oxidative phosphorylation. So it takes all these proteins embedded in the cristae, and then we've got this transport protein / enzyme that is where the actual phosphorylation of ADP occurs. So notice that oxidative phosphorylation is a lot more complex. And the substrate level phosphorylation does not require so many proteins and such a large structure. So it should kind of make sense that oxidative phosphorylation is very complex, and it can make a lot of ATP compared to substrate level phosphorylation. So even though it's more complex, you get more bang for your buck. And also notice here that there is no substrate, there is no molecule that is donating the phosphate group directly onto ATP. How the phosphate group gets here is beyond the scope of this class. If you continue to take biology in college and biochemistry courses, etc., molecular biology courses, you will learn how that phosphate group gets here to be part of this process. Now, I found another image on the internet, and this is an example of how you can get a trick question on this material, and you have to be careful to only see what you really see and don't let your brain put in information, because the person who made this image hates all of us. This is a mean person, okay? This person doesn't like us and wants to see us fail. Notice how they have the electron transport chain set up. Most of the time, you will see it drawn like this, where the inner membrane is on the bottom and then the outer membrane is on the top. And so when a very mean person comes along and makes their image like this, where it is upside down, then your brain is going to tell you that the top of the image is the inner membrane space and the bottom is the matrix. But if you look closely, this is turned... Oh my gosh, sorry. Luna, that is not something for you to bark at. Okay, sorry. She's letting me know that there's some monsters that I should be aware of. Thank you, Luna. Thank you. I appreciate that. I'm going to keep my eye on those monsters. All right. So let's get back to this person that hates all of us. This person is being tricky. And they may say it's an innocent mistake, but it's not. Most pictures are flipped. And this is a perfect example of don't let your brain put in your eyes what you're not really seeing. Clear your mind, look at the entire picture, and then you'll see in this upper left-hand corner, it says matrix. So you could easily get questions about this figure wrong because you didn't notice that they flipped it upside down. So I only have this here. You get older, you're becoming adults, you realize that there's mean people out there who are trying to trick us. So be careful. They'll do that with the Calvin cycle. They'll turn it around. The citric acid cycle, they'll turn around. And so you really, really, really have to be careful. All right. In class, we talked about redox reactions. And I found this video. So let's watch. Oops. Okay. Why isn't this working? Oh, control click. Oops. There we go. Now we go. Here we go.
Redox reactions. When magnesium combines with oxygen to form magnesium oxide, electrons on the magnesium atoms are more strongly attracted to the oxygen atoms than they are to their own nuclei, so they swap over, creating two ions. The magnesium ions have an electrical charge of +2 because they've lost 2 electrons, and the oxide ions have a charge of -2 because they've gained 2 electrons. Positive and negative charges attract, so the ions bond to form a lattice. This is magnesium oxide, a white powder. Oxidation is the loss of electrons, so in this reaction the magnesium has been oxidized. Historically, the only oxidation reactions that scientists knew about all involved oxygen. That's why they called them oxidation reactions. But now we know plenty of oxidation reactions that don't involve oxygen, like the formation of lead trees from zinc and lead nitrate solution. The zinc atoms have no electrical charge. Lead nitrate solution, on the other hand, consists of positively charged lead ions and negatively charged nitrate ions moving about in water. Zinc is more reactive than lead, which means it more readily gives up its electrons. So the zinc atoms lose their electrons to the lead ions. This turns the zinc atoms into positive ions, which move into solution, and the lead ions into lead atoms, which form beautiful metal crystals as they come out of solution. This is an oxidation reaction too. The zinc has been oxidized because it lost electrons. But one atom's loss is another's gain, and gain of electrons is called reduction. So with burning magnesium, oxygen is reduced as magnesium is oxidized. And when lead trees form, lead is reduced as zinc is oxidized. Oxidation and reduction are really just different aspects of the same thing, so we mix the words together and call the combination redox reactions. Redox reactions are reactions that involve the transfer of electrons. You can remember which reactants are oxidized and which are reduced using the mnemonic Oil rig. Oxidation is loss and reduction is gain. Oil rig.
All right. So they make a lot of really good animations and videos. And in cellular respiration, there are going to be redox reactions throughout the entire process. So questions you can get on the test would be to ask you which molecule is oxidized or reduced, or what is the reducing agent, what is the oxidizing agent. And in cellular respiration, as a glucose molecule is being broken down, those are oxidation reactions. When we learn about photosynthesis, we'll have a lot of reduction reactions being the main reaction. Yeah, I think that's all you need to know about that. All right, so facts about cellular respiration. There's a lot of factual information that you could be asked on the test. Then, of course, there's going to be a lot of application questions, which we're going to talk about at the end. In general, the kinds of facts that you need to know about cellular respiration are: what are the stages? What are the reactants? What are the products? Is CO2 released? Is ATP produced? And are the electron carriers involved? That is what we will be looking for and paying attention to. Once you know all of those facts, then I can give you some ideas of how that can be an application question. But you have to learn the facts first. So what we're going to do, we're going to watch a very good animation by McGraw-Hill. They make a lot of very, very good animations. And this animation will be all of cellular respiration. I want you to get an overview. Then we're going to go through each of the 4 stages, and we're going to discuss where it happens, where are the starting materials, where are the products, is ATP produced, is CO2 produced, are the electron carriers there, and is there something special that happens? And then at the very, very end, we're going to watch this animation again, and you'll find that even if you're not trying to learn the information, just by even watching the animations here and there over and over again, you start to learn it. Your brain starts to pick up the information. So not even trying to study at all, you can become familiar with the information. And then it's easier to learn, easier to memorize. So familiarity is what we're shooting for. And then by the time you have finished studying for the test, you will know the information better. So you kind of have to get used to not mastering the information immediately. You guys are all very, very smart. You can master information fast, like maybe even during class time. You hear it once, you got it, you're good. But now you're learning more difficult information, and then just a lot of information. So all it means is there are just extra steps now. You're still going to master the information, but now you have to take extra steps before that. So part of that is learning how to learn a lot of information. And this is one method. And I've told you before, pay attention to your teachers and your professors in college. They will all have different methods. And some of those are going to resonate with you and some won't. So you really just need to pay attention to see what works for you. All right, so let's watch this.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use.
Right. Now, you may have noticed that she called the last stage of cellular respiration the electron transport chain. And in class, I told you that that is common, just to call the whole thing the electron transport chain, when technically part of it is the electron transport chain. So it's annoying, but it's just something we all have to live with. Technically, oxidative phosphorylation is the name of the last stage, and the electron transport chain is part of it. And on your test, we will treat it that way. Your textbook says that the last stage of cellular respiration is oxidative phosphorylation, and that it's made up of two parts, one of those being the electron transport chain. So we will also use that nomenclature. All right. Part 1 of cellular respiration is glycolysis.
ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP, and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis.
She's going to go into a lot of detail that you don't need to know. In college, you actually probably not in the first bio class anymore, but if you take more than the first 2 biology courses, you would need to know the names of all of these molecules. So really, I just want you to, it's good to see the reactions, that it really, that it is different molecules and not these blobs and circles and triangles. So just know that you don't have to memorize that.
And focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
All right. This group also makes a lot of really good videos. All right, so we learned that glycolysis occurs in the cytoplasm. The starting materials are glucose. The products are 2 pyruvate molecules. We saw that ATP is produced here, and 4 molecules of ATP in total are produced. However, 2 are consumed, so there is a net of 2 ATP produced during glycolysis. There was not any CO2 produced during glycolysis. We saw that the electron carriers were involved, and it was the electron carrier NADH. Special information for glycolysis would be that it is in the cytoplasm, and it can occur all by itself without the other steps to produce a little bit of ATP, which is great if you're single-celled, but for bigger organisms, you would want to go through the whole process. All right, pyruvate oxidation is the part of cellular respiration that moves all the reactions into the mitochondria.